We've emphasized a lot in the course about genomics and genetic changes, but in cancer, another key factor is the silencing of tumor suppressor genes, and this can lead to malignancy. So I used to be a professor at George Washington University, and I met Mukesh Verma, our first speaker, at the university. And subsequently, in 1993, I came to the Division of Cancer Prevention and Control, and he came a little later to the Division of Cancer Prevention. And then I ended up in the Center for Cancer Research, and now he's in the Division of Cancer Control and Population Sciences. So he's the chief of the Epidemiology and Genomics Research Program, and he's going to talk to us today about epigenetics. Mukesh.
Thanks, Terry, for the introduction. Can you hear me all? Yeah. So today, as he mentioned, I will discuss about epigenetics. Now it is not as a new area, but a few years ago it was. So I am in epidemiology and genomics research program. So time to time I will tell that how our program is involved and we want to utilize epigenetics by supporting different cohorts. And cohorts are selection of different people. When disease has not developed, then we collect samples and follow them. What disease has come, so epigenetic markers along with genetic and other things also we follow. Cancer, either cardiovascular disease or cancer, they are either number 1 or 2 diseases in US, and 4.24 million men or 5.31 million women, they are affected by that. In men, prostate cancer is the main leading cancer. In women, it is breast cancer. And it is estimated that with the time, number of cancer cases will be more. Although we have succeeded in some cancers, like colon cancer by screening or breast cancer, but in cancers like pancreatic cancer, ovarian cancer, we do not have early detection markers. That's why we want to put our lot of efforts in identify such markers which can be used either for risk assessment or can be target later on for therapy. So cancer, major cancers like breast, cervical, colon, prostate, lung, all these, they don't develop in 1 or 2 years. They take long time, 15, 20 years or 18 years. So our whole idea is that if we can identify some markers at initial stages, then it will be good. We had more options for therapy and other interventions. For example, in colon cancer, where maximum work has been done, so it has been identified that with age, at different age, different markers or different histological characteristic features come that we utilize. So based on age or different stages, pathological molecular biomarkers that we want to utilize, but whole idea is that as early we can detect it, that is the best. So since our most of the biological information that has come from genetics, so and we want to utilize whether we are doing epigenetics or others, whatever information we have, how we can use that. So few years ago, after genomic sequence was done, then GWAS, genome-wide association studies were done, where idea was that cancer-associated genes or location, if we can identify, that will be good. So in 2005, 1 or 2 came. Then as time passes by, on different chromosomes, as you see, those genes were identified where genetic alterations, they are associated with different kinds of cancers. And now, in many cancers, these genome-wide association studies have been completed. So in cancer, at least we know for different kinds of cancer, like as you know, in different organs, cancers are different. What mutations commonly happen, that we know. Similarly, we know what are the major pathways based on genomic analysis and others. So that information always is in background, but environment or lifestyle and other features are such which also affect and contribute to cancer development. And in that concept of epigenetics come. So what is that epigenetics? changes in gene expression without changing genomic sequence or genomic structure. So you know about genomic code, but methylation code and histone codes are also there. In general, you can imagine that what you are capable of, that is genetics, and when that capability or when you want to use that, when you want to stop that, that is epigenetics. And as the name also implies, epi means above, so it is above genetics. So to distinguish epigenetics and genetics, a lot of studies were done in monozygotic twins where genetic background was the same, but still discordance was followed in those twins. And then it was realized there is another science also, and then epigenetics, that's how it came. And time to time, popular articles also come, like in Time it came that your DNA is not your destiny, but choices you make, and you can change your genes. By changing lifestyle or environment and other things, they are also equally responsible for disease development. And in my talk later on, I will be covering some pharmacoepigenomics versus pharmacogenomics, or how much microenvironment or microbiome affect epigenetics, and the way we do GWAS or genome-wide association studies. Similarly, I have put some reviews in epigenome-wide association studies. Now, along with cancer, in some behavioral aspect also, epigenetics has been shown that works, and you can see that epigenome-wide association studies for behavior. So it is expanding, and now almost whatever diseases are there, some or the other, evidence are coming that epigenetics regulation is involved in that. To give you a little background, nucleosome are units of our chromosome, and it has histones or non-histone proteins. Histones are of 4 kinds: H2A, H2B, H3, and H4. H1 is just to link there, and the reason these proteins are needed to neutralize acidic part of DNA. And what are the major components of epigenetics? First comes methylation part. What happens that in the promoter region, some CG sequences are there. In cancer, they get methylated. So methylation is one part where C gets methylated. And different enzymes have been identified for methylation. They are methyltransferases, DNM, T1, T2, T3, like that. Some are to initiate methylation, some are for maintenance of methylation. Environment, hormone, genetics, all those affect epigenetic changes. And epigenetic regulation almost in all steps of cell cycle regulation, you can see that. The way chromatin is arranged in cell, either it is compact or relaxed. So in compact position, transcription factors cannot come. In open position, they can come. And some other non-histone proteins like polychrome group of proteins have been identified, or certain those have been also identified. Histone proteins get methylated at different sites by acetylation, biotinylation, methylation, phosphorylation. Different kinds of these are there, and they work together. For histone modification, different enzymes have been identified for methylation as well, and little bit about its clinical implication I will be telling later on. So as I mentioned, methylation of DNA, modification of histones, and later on it was observed that non-coding RNA or long non-coding RNA link and microRNA, they also come under epigenetic regulation as well as genomic imprinting. So if in promoter region more than 40% CG sequences are there, then when cancer starts developing, then tumor suppressor genes get inactivated, and as it is shown in red, that CG cytosine is converted to methylated cytosine. At the same time, which has been observed in oncogenes, their promoters are already methylated, but they undergo hypomethylation, and thus oncogenes are activated. So both ways it works. And now for different cells or different kinds of organs, these are very much distinct. Now these are different factors which affect genetics and epigenetics both. And when twin studies were done for different diseases, then some diseases like neurological disorder, schizophrenia, Alzheimer's disease, and all that, in that genetics is a major component. But in cancer and some other diseases, epigenetic components are more. And lifestyle and other diet, etc., are such which you control, then you can control epigenetics also. So now little bit demonstration here. It is a nice video I like that will give you more background.
There are over 100,000 skin cells in 1 square centimeter of skin. Each of these cells contain close to 2 meters of DNA, which contain our genetic information. All the genetic material in our body is called the genome. Because our DNA is very long, cells package up their DNA together with proteins known as histones. The DNA and protein complex is known as chromatin. DNA is wrapped around histone proteins, forming repeated units of nucleosomes that look like beads on a string. Chromatin is condensed further to form a chromosome. We have 46 chromosomes in total, stored in the nucleus. The core of one nucleosome is composed of DNA wrapped around a histone octamer that consists of 2 copies of the major types of histones: H2A, H2B, H3, and H4. This organized DNA protein complex allows the cells to regulate what genes are expressed and when. All the cells in our body contain the same DNA sequence. Skin cells, muscle cells, and liver cells contain the same DNA sequence. Yet all these cells have different structures and functions. The reason is that different cells only use or express certain genes. Within each cell, the DNA and also the histones can be tagged by tiny chemicals that modify gene expression. These chemical tags cause some genes to be turned on and some genes turned off. For example, muscle cells will have genes turned on that a muscle cell requires, and will turn off genes that a liver cell requires. Similarly, the liver cell will have genes for muscles turned off and genes that a liver requires turned on. These modifications are known as epigenetic modifications. Epi is Greek for above the genes, so modifications above the genes or on the genes. Epigenetic modification brings about lasting changes in gene expression. Let's have a closer look. Here is a normal human cell, and we are pulling out its genetic material. The DNA is wrapped around nucleosomes, which are made up of histone proteins. Coming off the histone proteins, we have histone tails. DNA is made up of a combination of 4 nucleotides: G for guanine, C for cytosine, A for adenine, and T for thymine. There are 2 main epigenetic modifications. The first one we will talk about is DNA methylation, where a methyl group is added directly to a cytosine residue that exists in a cytosine guanine sequence, or CpG sequence for short. So for example, here we have many CpG sites that make up a CpG island. And here the cytosine are methylated. The methylation of CpG sites in promoter regions is associated with gene silencing. The second type of epigenetic modification is histone modifications. This is where acetyl or methyl groups are directly added onto histone tails, and this will modify gene expression. For example, histone 3K9 acetylation correlates with transcription activation, and histone 3K27 trimethylation with transcription repression. Genome-wide patterns of DNA and histone modifications, or epigenome, are established during early development and are maintained during cell division. In cancer, these patterns are altered and disrupted.
So that was little background I wanted to give to clarify how does it work. Another point I want to make, because not only for cancer but for other diseases also it applies, that at early stages, whatever changes are there, so chances of error due to different exposure or risk factor, that is more at the early stages based on the weight or number of cells and all that. So these are the very high chances at early stages and later on low. Same way, if you see here also that diet and lifestyle, those changes are at initial stages, and at the later stages and initial stages, these are maternal factors and other hormonal factors. So another objective of showing this slide is that at different stages, biospecimen can be collected for analysis. So that way you can do analysis, whether it is histone analysis or methylation analysis. And so during development, these steps, in our one of the program at NIH Epigenome Roadmap, those steps and different changes have been characterized. So you can consider if a mother is smoking, then 3 generations are affected, mothers as well as a child and the stem cells in child. So that's how it works. So early life exposure, that is so important. In environment, different kinds of minerals and other compounds are there. Originally people used to study only DNA breakage and other things, but epigenetic modifications are also affected by that. And some of these environmental chemicals and other things I have shown here. Whenever changes are for short time, like cell cycle regulator, transcription factors, so anytime exposure or risk factors are there, they will affect that. When it is little longer, then epigenetic changes happen, and when it is for really long time, then only genetics start. And nowadays, since we know about microbiome also, so that comes in 3 or 4 months, people's microbiome also change. So all these things you have to keep in mind. Suppose your study involves very quickly on short-term basis you want to study, then you have to focus on this part. And if long-term basis you have to do then on other parts. In different cancers, about genetic background, translocation, deletion, amplification, indels, and other substitutions have been characterized for different cancer. Same thing we have done for epigenetics to a certain extent. Peter Jones and Andrew Feinberg from Southern California and Johns Hopkins, they are the pioneer in epigenetics field. And they actually identified that first activation of acetylation by S3 is that histone, and lysine 9 position that happens, and then inactivation by deacetylation, then silencing by attachment of one methylation, and then only DNMT comes. And on this slide, it has been shown that anytime if nucleosomes are separated from each other, then acetylation is there, no methylation, and transcription factor can work. So it is like euchromatin, but gradually methylation starts or deacetylation. Because of that, chromatin gets complex and gene gets inactivated. And this is a sequential phenomenon. So whenever you want to plan any methylation project, then both strands you should sequence, because that goes one strand first and another strand later on. Generally, for most cancer, it is true that global hypomethylation happens, like line sequences and sign sequences which are repeat, they are already methylated. So they undergo hypomethylation, means removing methylation, but promoter region gets hypermethylation. Similarly, histone modification also as the cancer or the disease progresses. In methylation also, we have either abnormal increase or decrease or no change. When increase is there, as I mentioned, methylation of both alleles, or one allele, another mutated, or one is hypomethylated, another by recombination, or in decrease also, like proto-oncogenes. When gene therapy came, at that time, many times recombinant vectors, they did not work. The reason was that their promoter was LTR, and which got inactivated by epigenetic mechanism. So in that also, a lot of things have been shown by abnormal decrease. And no changes, that is by chemically, for example, deamination and other processes are there. There also methylation is involved. Now, mutations affecting methylation profiling. After genetics came and epigenetics also, we knew few changes. Then people started checking what mutations exist and whether they are affected by methylation or not. So these are few cases where systematic studies have been done, but that lot more has to be done. Another part was also seen that epigenetic regulation in whole pathway at different step has been characterized. For example, integrin signaling pathway. microRNAs. 8-10 years ago, we did not know whether humans have microRNAs or not. They were in Drosophila or plant and others. Then in human they came. After human, they were tissue-specific, and now we know cancer-specific microRNAs are also there. The way we utilize in epidemiology, either whole microRNA profiling you can do, or those genes which are coding for microRNA, those can be followed whether some changes are there which are associated with different cancers or not. So they are very useful. Anytime we deal with RNA, that is very labile, but microRNAs, because of their size of 21 to 26 nucleotides and secondary structure, they are stable, and different kind of biospecimens you can use either for risk assessment or diagnosis or follow-up of treatment. So microRNA polymorphism also has been utilized for identifying high-risk population. And here it has been shown that microRNA work both ways, either they can increase activity of a gene or decrease. So in different systems, they work differently. In histone, as I mentioned, that 4 kinds of histones are there. These are different modifications which are cancer associated. So there was a problem 7-8 years ago that if someone wants to characterize these modifications, they did not have high quality antibodies. So NIH initiated one program, it's called Roadmap Epigenomics. And in that program, we proposed that first of all, profiling of healthy cells in terms of methylation, microRNA and histone, and chromatin compactation by DNase-1 profiling, that will be done. And whatever modifications are disease associated, so these monoclonal antibodies will be raised by contracting to some companies, and those monoclonals will be distributed so that people can study not only in cancer but others also. So that's the reason of showing this slide. Now, few terms you will be reading about readers, writers, or erasers. So that those are used for histone modifications at different times. Either some additions are there or some are removed. So those are different terms which you will be hearing. And technologies are such that at different location you can follow time-wise or temporarily that what are the changes in histone. So those things are possible now. Few examples are shown here where different modifications in histones at different location, either they activate or inactivate gene. And to promote this field, time to time I have edited books. So first book I did, that was the first one on epigenetics and cancer prevention. Then 2 I have done on cancer epidemiology, one for host susceptibility factors, one is on risk modifiable factors. In this, like alcohol, environment, radiation, or infectious agent that you do, and in host susceptibility factor, how your background of genetics or immunology that is affecting epigenetics. And this was the second edition where how we can utilize this for risk assessment or epidemiology. And cancer biomarkers I did in this epigenetics marker, how early we can detect what is the current status. And this one is coming next year. One time I was interviewed when epigenetics was coming, that how it is different from genetics and why do you promote that so much. So Nature interviewed, and then I mentioned that epigenetic changes are reversible and therefore have an edge over genetics. If you have some mutation or other changes in genome, at the most you can tell, okay, this disease may be developing, or you can follow up that for diagnosis, but you cannot go back. But epigenetic changes which I mentioned, since their genome does not change, so you can reverse. And few examples I will show there. So that is the strength of epigenetics. Next year again I was interviewed by them, and that was after that I mentioned that successful approval of first generation of drugs intended to target epigenetic pathways has convinced almost every major drug company to invest in cancer epigenetics. So it started with leukemia and other cancers, but in other diseases also, 6 compounds FDA has approved, which are either histone deacetylation compounds or methylation. So histone inhibitors or methylation inhibitors, those have been approved by FDA. So if some methylation project is done, then 4 or 5 things are kept in mind: methylation content, methylation levels, pattern, as well as profiling and pattern profiling. And this will reduce your false negative and false positive results. Steve Bellin, who is in Johns Hopkins University, So he has identified different steps when different proteins in a sequential way come to the promoter and how we can target those. So he has used few epigenetic drugs which has reduced lung cancer also. So those kinds of papers are there. So if in PubMed you search with Steve Bellin and epigenetics or translation, so you will see those results. And these kinds of things have also identified that if lysine 27 gets trimethylated, then it will affect differentiation. Or if K9 or lysine 9 has monomethylated and next amino acid is serine, then upon phosphorylation, changes will be permanent. So those kinds of characterization Steve Bellin has done. So a lot of basic things have also been done. And during tumor development at different stages, different samples from exfoliated cells have also been identified. That is just to determine whether cancer is progressing or not, or for follow-up of treatment as well you can use. On this slide, I have shown some of the selected group of genes which in different cancers, they are affected by methylation, because for first time of screening, they are very useful. At the same time, for histone deacetylase or histone DNA methylase or histone transferase, different kinds of different genes are affected, which have been characterized very well. So those are shown on this slide. These inhibitors, whether for DNA methylation or histone, few of them are shown here. They are at different stages of phase 1, phase 2, those trials. And Jean-Pierre Issa, he has been involved in very much in SAHA, one of the compound which is giving promising result. He has done a lot of work. Although some of these compounds, like it happens with any agent or any drug, some kinds of side effects are also there, but we follow how much are the benefit and how much are the harmful effect. That way, investigators, that way those results or those studies are modified. So different inhibitors in different categories like HDAC inhibitors or histone transferase inhibitors, their commercial names are shown here. And companies like Johnson & Johnson or AstraZeneca or Merck, SmithKline Beecham, all major pharmaceutical companies have program in epigenetic drugs. So this is coming up very well. And the whole idea, as I mentioned, that is that these changes, if you detect early or at certain stages, you can bring that back. Few examples I will show, in which cases those patients were taken, cancer patients, who were not giving response to any kind of therapy. So in that, in combination, methylation inhibitor as well as histone inhibitors, they were given. And at different days, like 1, 10, 28, histone modification and methylation profiling was done, and they started giving response. So success was there. At the same time, 5-azacitidine was combined with valproic acid, which is for histone, and all trans-retinoic acid. And in that also more than 50 patients that they were involved who were not giving any other treatment response. So in that also we were successful. And if you see on clinical trials website and look for histone inhibitors, then several trials are going on. And these are not only for leukemia, but for solid tumors also of different kinds, they are going on. And if you go on that clinical website and click the hyperlink, you will know how many pupils are enrolled, who is the principal investigator, what are the objectives, how many are giving response, what are the future plan. So if you want to know the clinical implication, this is a very good site, cancer.gov, and within that you put clinical trials. Or in Google also clinical trials at NIH or NCI you can put. So these are FDA approved epigenetic inhibitors, 2 for methylation, 2 for histone modifications, and few more were identified. So this slide I updated. So now we have 4 in histone and 2 are in methylation. So this number is also increasing. And as you know, FDA generally takes 7, 8, or 10 years by the time it is approved. So you can imagine how much work was done, and then only we came up to the stage that these can be used. And this is one of the paper where non-small cell lung cancer, in that treatment was done using these inhibitors. We call it epigenetic therapy. Similarly, for hematological malignancy also, that has been used. Now, in cancer, in 15 to 20% of cancer infectious agents are involved. For example, if you see papilloma virus in cervical cancer, gastric cancer is H. pylori, liver cancer, HBV, HCV, and all that, and then HHV8 and others for Kaposi sarcoma. So those are such that they have been involved in cancer. So then scientists started studying about epigenetic regulation. So two genes like latency associated nuclear antigen or Epstein-Barr virus for nasopharyngeal carcinoma, that antigen, those were characterized. And then in 2009 for HPV, EBV, and HBV, whole methylome was characterized in these viruses. Why we do those kinds of things? Because many times we don't know any pathological, histological, or other molecular markers by which we can tell who are the people developing these different kinds of cancers. But by doing these molecular markers we can use for predicting that who is at high risk of developing cancer. So this is one example for HPV 16. So early genes or late genes have been identified, but at the same time, which gene will be how much methylated, those characterization are done. So these we use as biomarker. And also in biomarker field, general concept is that anytime for risk assessment or diagnosis or other purposes, don't use single markers. You take patient information, risk factors, lifestyle, genetic markers, others, and then see how much is the sensitivity and specificity, and then you predict. So along with methylation, other things are also taken, and then based on like SYN1, SYN2, SYN3 are in cancer, cervical cancer, that we do. Since epigenetic changes can be modified by drug, but it was also observed in, we call it nutritional epigenomics. So in that some nutrition or food as such, or natural food, or their bioactive food components, they have either histone deacetylation properties or DNA demethylating properties. So this is also recognized in cancer field that by changing lifestyle, diet, and other things, 30 to 40% cancer progression can be reduced. In smoking we saw, as well as screening of breast cancer that we saw, colon cancer, finding polyps, that as well we saw. So in this example, methyl deficient model was developed for hepatocarcinogenesis, where only methyl donor methionine or other things were changed, and such cells were produced which are normally in animal model, that was characterized. And these are some of the natural foods in which either both activities are there for DNA methylation or histone modification, or only for methylation, or only for acetylation. We have one center of alternative medicine. So in that, few studies are going on. Otherwise, in extramural field also we support such studies. So about NIH roadmap I was mentioning. So when Dr. Zahroni was our NIH director, so he decided that certain mechanisms are such which can be applied for many diseases, not one diseases. So they dedicated about 1.2 to 1.5% of NIH budget and invited advisors that which area we should develop. So at that time, microbiome and epigenetics were approved for that. And as I mentioned in epigenomics, we wanted to do the way human genome was completed and human proteome was completed. Similarly, we wanted to do in epigenome also all those 4 components. So that was approved, and that time it was called roadmap. Now after Francis Collins has come, so its name has changed to Common Fund. Idea is that some area is developed, and then institute can support that research based on what disease they are focusing on. And in epigenomics program and another metabolomics program, and third one is molecular transducers of physical activity. These are 3 common fund programs where I represent NCI. So from the initiation up to date, I have been involved in that. So by this program, we wanted to follow up for 5 to 10 years with big money that high risk kind of scientific field in which people cannot get grant or projects are not sanctioned, that we should do, which is transformative myths, which has clinical implication, and this will be trans NIH. Whether some institute participate or not, money will be taken out from that institute to support these kinds of projects. So just to give you a brief idea, during these years, like 2007 or 6, it was started. Now in 2015, we spent about $500 million on this. And these are different programs which are ongoing. So as I mentioned, I am in epigenomics. Microbiome is also there. Extracellular RNA communication, like extracellular vesicles are there that you can target for different reasons. Single cell analysis, so some are basic science based, some are typical that clinical implication, and some are bioinformatics oriented also. Like BD2K, when you hear about that, that is supported by Common Fund. Epigenomics, if you want to see that 5 major projects were started with 219 million committed dollar. Like some mapping centers were identified where they can map methylation changes, microRNA changes, histone changes, and DNase 1 mapping. Then disease-specific projects were also developed, and as technology changes time to time, so those centers were different. And these were such that investigators from all over the world or US, they can take their help. So some money was given for collaboration. And after epigenome was completed, then in Nature few articles came, and in that all protocols or assays or what are these changes, those were published. And as in that presentation, video presentation in my talk, I mentioned that genome, when you sequence, since genome is the same whether you take from skin or liver or eye, anywhere, DNA is the same, but epigenome changes. So before we started epigenome, we knew about that. So 112 kinds of cells were identified, and in that, in all of them, epigenome, all those 4 components were done. So all that data is NCBI and dbGaP. So when people study any disease, and here samples were taken from healthy people, like pathologically healthy people, and from that samples were taken. So that is a reference point to compare for any disease, whether it is diabetes or whether it is upper airways or heart diseases, cardiovascular or cancer. So this data has been very useful. After we started Roadmap Epigenome, then International Human Epigenome or IHEC program was started by Europe, and I represented NIH in that. So in that, they decided that they are going to do 1,000 epigenomes, the way 1,000 genome idea came, or if you are keeping track about personalized medicine initiative in which 1 million people will be followed. So that kind of program, and here they also wanted to do reference standard and in stem cells, but along with that, they wanted to see what are the age-related changes and what are the disease-oriented changes. And this is the website for IHEC. And in first meeting, which we had in 2010, so these are the countries who participated initially. Now more number of people are there. And the way Europe organized, that countries have to commit at least 25 million or $30 million to join that group. And then data or resources can be exchanged, but this they are expanding. And other agencies also joined after they started in Europe. And companies like Affymetrix, they provided their equipment to do the analysis. Genoscope, Novartis, those were involved. And that time, all coverage was done by Nature and Science about this IHEC program. So this is the actual website. Annually, we meet for this IHEC program, but for Roadmap Epigenome, every 6 months face-to-face meeting, but every month we have steering committee meetings, because in different diseases now, this epigenetics is being involved. So these are the new members of IHEC. And in our epidemiology or other things where we feel the research opportunities are there for our program, one of them is inclusion of epigenetic markers, whether we can identify new risk factors. Few we already know, but few more we want to identify. Because originally people used to take just lifestyle factor or family history and others. And then in cohort case and control studies, in that whether we improve sensitivity and specificity also, if we include these to identify high risk population. How much they are correlated, that is also still going on. And in that, after GWAS studies were completed, then everybody expected those SNPs which are associated with those changes, they should come, or they should identify genes which are affected. But most of the SNPs came outside the coding region. So then also that suggested that genetic regulation is not the only one, we have to follow others. But only in few cases, not in cancer, but in other diseases, those SNPs are in coding region. So in that, along with genetics, epigenetics is also being explored. Race, ethnicity, and microRNA, how much they are correlated, that we want to know. How we can define cancer subcategories. Pathologists, most of the pathologists are trained all the time. They cannot distinguish, like in breast cancer, we have ER negative, PR negative, or positive, or triple negative cells, and one cells will respond to treatment, another will not. So such kinds of things in that molecular characterization, in that also this kind of information is very much needed. And epigenome-wide association studies, the way genome-wide association studies are done by identifying locus and SNPs, same way we say that all these 4 markers, chromatin markers, methylation, histone, microRNA, and DNA is one mapping, that we use in the same sample. And in one of the program, Common Fund program, which I mentioned, molecular transducers of physical activity, in which simultaneously we are measuring that. So that program was out just 5-6 months ago. We have identified investigators. Idea is that one healthy group will do physical exercise, another will not do. From that, different kinds of samples, biospecimen will be taken, and transcriptomic profiling, genomic, microbiome, epigenome, and metabolomics. All those will be done, but from the same person, so that individual variation is less. So that kind of programs are also going on. So epigenome is part of that. Now, based on all these molecular analysis, so much evidence was accumulating, and whenever you give a drug to 1% or to 20% or 30%, few of them will not respond to the same drug. It means their background is important. Now, in pharmacogenomics, they identify what is the basis of genetic background, which is making them either responsive or not responsive to treatment. So pharmacogenomics came. Then now epigenomics is also there. So President Obama initiated this precision medicine initiative, in which 2 institutes were funded, more than 200 million each, NHGRI and NCI. So in next few years, about 1 million people will be followed at different stages, what disease they develop, whether some marker comes or not. So that was a big initiative about precision medicine. Now, precision medicine, since that name came, so recently, just 1 month ago, its name has changed to All of Us. So you will not hear about PMI, but that name has changed to All of Us. That was declared. Now, a little bit about my program, how this epigenetics and other like GWAS studies we promoted, how do we do that? So we have different cohort, and cohort have consortia. Again, to emphasize, cohort is a group of people where disease has not developed, but they are followed and biospecimens are collected either every 6 months or 1 year or 2 years at different times. Some of the examples are nurses' health studies, physicians' study, multi-ethnic cohort studies, or Chinese studies. So all over the world, Australia, Japan, India, China, and Hawaii, Puerto Rico, all those places, we support those clinicians who are seeing patients or who can collect data. And that we keep in one place. So if somebody designs some experiment, they can utilize that data or some specimen to further characterize. Idea is that, first of all, we want to understand etiology, means what are the factors which are contributing to disease development. Once we identify those factors, then we think about how we can prevent them or treat them, although that goes for division of cancer treatment. So these are some of the examples of these cohorts. And on our website, these are all hyperlinked. You will know who are the people collecting the data, what kind of results have come. And most of the GWAS studies have been done by our program. And epigenomics also, about 63 projects we have. In my portfolio, about $72 million we spent on epigenetics program. So that is a major program. And if you want to sort out for different cancers, like your pancreatic cancer or others, you can identify what studies, epidemiological studies are going on. So most of the studies are to identify risk factors, but one of our branch in the program is, it is called clinical epidemiology branch. In that, we want to find out after the treatment, cancer treatment, how they are doing, those patients are doing. Because in epigenetics inhibitors, what was observed, that sometimes they work as radiosensitizers. So if you treat with epigenetic inhibitors first and then use regular cancer treatment drug, then patient start responding. So combination therapy and this therapy, in that epigenetic inhibitors are being used. So in conclusion, I want to say that epigenetic changes are present in all human cancers and are now known to cooperate with genetic alteration to drive cancer phenotype. These changes involve DNA methylation, histone modification, readers, modelers, and those erasers I mentioned, and then microRNA and long non-coding RNAs and other components are also part of epigenetic regulation. And to identify different malignant phenotypes or identify different subtypes, these studies are very important. And genetics is our basis, but we have to go for alternative also, because after researching for 30-35 years and keeping all focus on genetics, good results have come, but they are very limited. And now we know that different people respond to the environment and lifestyle differently. So this provides us very good opportunity to study epigenetics. And the use of epigenetic therapy in sensitizing treatment with immunotherapy has been explored. So in immunological cells also epigenetic studies have been conducted. The main challenge which comes, since in all cells epigenome is different, then how to follow that and how to make target specific. But somehow such observations are only now, they are existing where treating with these agents, benefit results are coming. But to understand or to regulate at a specific organ sites or specific cells, more work has to be done. Epigenetics provides direct and quantifiable link between environment influence and relevant pathways. So NIEHS, another institute, National Institute of Environmental and Health Science, they are also very much collaborating with our Cancer Institute. And longitudinal studies should be planned to implement epigenetics for personalized or precision medicine. The reason is many changes happen. Suppose one time only you take reading, then next changes or in between changes you may not know. So same patient with the same doctor should be followed for a long time, or if electronic health records are available to doctor, they will know about that. And that was also basis of personalized medicine. So that and for initiation of All of Us program also, that was the basis. So I think that will be the way that patients should be followed longitudinally or at different times they should be followed. And since we have that sister program of metabolomics, and metabolites are easy to measure in blood or urine, so that also we encourage that if you are doing genetic studies or epigenetics, since you are collecting samples, so you can do metabolomics also. And to encourage different investigators, what we say, that suppose in your proposal or you are not planning metabolomic studies, then from our NCI we can put some administrative supplement or additional money so that people can follow metabolites also. So that way we want to bring different molecular markers together and see how good they are for risk assessment and identify different factors. And these are some of the collaborators who have helped me a lot. Thanks a lot, and I will be delighted to take questions. Yes, first that on your back also, later on. Yes. Yeah, so as I mentioned, this is a challenge, but in few cases what has been observed, especially Steve Bellin. So he was following non-small cell lung cancer and the tumor development. So patient initially came, they tried with the treating with different things, it was still increasing. So they were taking several images, and after they treated, so they have not checked different organs, but at least that tumor was reduced and health was improved. And those clinical trials also which I showed, so at initial stages, those patients are enrolled who have cancer and who are not responding to treatment. But still it is a problem. Suppose we want only it should go to lung or target specific we want to make, so that is still a challenge. Yes, Parvez.
Is there any microRNA focused program in terms of treatment?
Yes, so some our program supports some grant in which we have identified not LET7 but 121 or few others selected mostly in colon cancer, but some in liver cancer also. They want to show that target is specific since some sequence they have identified. Mostly they are for risk assessment. So initially they found in 30, 40 people. Now they propose in 400 people they want to see whether same kind of modifications are there or not. And then they want to attach some drug, or in the recombinant they want to put some drug along with that, and they want to put that. So partly they are animal-based models, some are in human. So human part we support and other part DCB support. Most of our programs are for risk assessment, for treatment also. In DCTD, some of them are coming. So since they are stable, that they can see. And also 2 projects recently we approved, and DCTD was part of that. In fact, I am part of one funding opportunity announcement or program announcement. Its title is Validation of Clinical Samples, or identifying biomarkers for clinical samples. In that, treatments are going on or treatments are proposed, but microRNA within exosomes, drug has been added to exosomes, synthetic exosomes, and microRNA are there, and then they are following whether that works or not. So exosome field is also coming. Like previously, only circulating cells people used to do, but they were not as stable as exosome. It has bilayer membrane, as well as it has sometimes DNA you can detect, but definitely microRNAs are there, lipids are there, and some other things are there. So that field is also expanding.
Thank you, Rakesh.
Very good.
So another class that we did this fall was redox biology, and this has to do with gases and their effect on cancer. And Perez lectured in that class, as did I. And I've lectured in TRACO, as you know, and now it's Perez's turn to lecture in TRACO. So Perez, he got his PhD in India, subsequently was a postdoc in Switzerland. He was on the faculty at the University of Maryland Medical Center, and now he's in the Laboratory of Human Carcinogenesis. He's in charge of the Pancreatic Cancer Unit, and he's going to talk to us about pancreatic cancer, current understanding and future challenges. Perez.
Thank you. Thank you, Terry, for the introduction. So what I am going to do today is to give you a brief overview about pancreatic cancer. What are the current progress? What is our understanding? And what are the challenges to improve survival in this most lethal malignancy in the world? So let me start with this slide, which simply shows that it's the third leading cause of death in the cause of cancer deaths in the United States. You see here it's fourth, but when you combine both men and women, it comes to the third leading cause of cancer deaths. The median survival is less than 6 months, estimated 53,000 new cases, and about 41,000 deaths will occur in 2016, and there is no effective treatment. So everything seems to be very disturbing, and the most disturbing is that according to the estimate, it is going to be the second leading cause of death due to cancer in the United States by 2030. So what are the risk factors for developing pancreatic cancer? These are some of the risk factors, for example, smoking, long-standing diabetes, pancreatitis, obesity, and also non-O blood group O. And these are some of the cancer syndrome that are inherited syndrome, where you have mutations in different genes, and that enhances the risk of pancreatic cancer. For example, hereditary pancreatitis, where you have mutation in the trypsinogen gene that enhances the risk to about 50-fold. So in terms of treatment, we have not made much advancement in terms of treatment. If you look here, about 20 years ago, gemcitabine, which is a chemotherapeutic drug, was approved over 5-fluorouracil because it enhanced the survival only by about 1 month. And then about 10 years later, erlotinib, which is a EGFR inhibitor, was used in combination of gemcitabine, which enhanced the survival only by about 2 weeks. But the situation is so desperate that it was approved by FDA, and it doesn't work in advanced cases, and maybe there is a small subset that will respond to these combinations. And then in 2011, a combination of different chemotherapeutic drug, and the combination is called FOLFIRINOX, that has the 4 different chemotherapeutic drugs that responded well, relatively speaking, in pancreatic cancer. As you can see here, it enhanced the survival in advanced cases, 11.1 months as compared to about 6 months by gemcitabine alone. But the problem was that it is highly toxic for majority of the patients. And then recently in 2013, albumin-bound nanoparticle paclitaxel was approved in combination of gemcitabine, which enhanced the survival for few months as compared to just gemcitabine, but it is less toxic than FOLFIRINOX. And then most recently, last year, nanoliposomal irinotecan and other 2 chemotherapeutic drugs were approved, and this also enhanced the survival by about a couple of months when used in combination, and it has less toxicity. So as we know that the dismal outcome is because of the late diagnosis. There is no early detection marker, and I will show you 1 or 2 slides about the situation in terms of early detection, and very poor therapeutic response, as I just mentioned. But there are about 18 to 20% of the patients that are detected early, and they are detected early because they are examined for something else, but the physician finds that, okay, you have the earliest stage pancreatic cancer, and these earliest stage pancreatic cancer patients can qualify for surgical resection. So you will imagine that, okay, stage 1, it is resected, everything is fine, but still the median survival, even in these 18 to 20% of the patients, is less than 2 years. And so majority of the patients, about 80% of the patients, show recurrence of the disease within 2 years. But there is a small minority of patients, resected patients, that do survive for 5 years. So are there differences among these early stage patients? Are there molecular differences? There are several clinical prognostic factors, for example, stage, grade, lymph node spread, and resection margin at the time of resection, that suggest the survival, good or bad survival in these cases. However, we do see variable outcomes. Sometimes you see stage 1 resection margin 0 and the patient die within 6 months. But in some cases, the same stage, same grade, same resection margin status, they survive for 2 years, 3 years, 4 years, or 5 years. So one of the hypotheses is that there are molecular differences in tumors that actually determines patient outcome. So how we can improve survival in pancreatic cancer? We know that we don't have early detection marker, response is poor. So we need effective therapy, and for effective therapy we need novel molecular targets that can be effectively targeted. And then molecular subtypes, that molecular makeup of the tumors that will respond to a specific drug. And treatment selection can be done based on the molecular subtypes. And then drug delivery. So especially, and I will talk a little bit more about the drug delivery in pancreatic cancer. It's very difficult because the tumor is surrounded by a huge stroma, and it's kind of highly protected, and it's also hypovascular, means it's not vascularized well. So when you give a treatment, the treatment is not reaching the tumor because the less number of blood vessels, and also blood vessels are collapsed. So there are some experiments that have been done to reduce those interstitial pressure on the blood vessels, and that has improved drug delivery and also survival, which I will talk in a minute. And also we need early detection markers, the biomarkers that can in fact detect precancerous lesions. Because once the cancer is in stage 1 or a stage 2, it's already too late, as I discussed earlier. So we need to have biomarkers that can be that can identify patients when they have precancerous lesions. So for all these, we need to understand tumor biology that will involve both genetics, epigenetics, tumor stromal interaction, metabolic reprogramming, and transcriptional dysregulations. So let's talk about, in 1 or 2 slides, the biology of pancreatic cancer. So it is developed by well-defined precursor lesions, which are called pancreatic intraepithelial neoplastic lesions. This is the normal duct, pancreatic duct here, and this is the pancreatic intraepithelial neoplastic lesion 1. You can see here some dysplastic changes going on. This is grade 2, and this is PANIN 3, which is almost carcinoma in situ, and then it penetrates the basement membrane and it forms this glandular-like structure, which is called pancreatic ductal adenocarcinoma. And PDAC, or pancreatic ductal adenocarcinoma, is the most common form of pancreatic cancer. It's about more than 90%, 90 to 95%. And there are several well-defined genetic alterations that occurs here, like telomere shortening. KRAS mutation is found in about 90 to 95% of the patients. Then P16 alteration, both by epigenetic alterations and mutations, and also P53 and SMAD4 mutation that are found in about 50% of all the pancreatic cancer. As I mentioned, this is just a section of the pancreatic ductal adenocarcinoma, which shows huge stroma. These are the glands, pancreatic tumor glands. And then here it shows that there is a strong interaction between tumor and stromal cells, and they support each other. So as I mentioned, there is a lack of any marker that can detect this cancer early on. There is only one marker that is clinically used these days. This is carbohydrate antigen 19-9. So anything in the serum, if it is more than 37 units per ml, that is a marker of pancreatic cancer. But the problem is that sensitivity is only 80%, and specificity is also not that good. So this is not a very good marker. And it's also not just specific for pancreatic cancer. And also, if you compare the malignant versus the benign pancreatic disease, both the sensitivity and specificity is very low in this cancer. So one study that was published last year by Raghu Kalori's lab, what they have found in the circulating exosomes, if they are glypican positive, glypican is a proteoglycan, and they found that majority of the pancreatic ductal adenocarcinoma, they have circulating exosomes that have that are glypican positive, and this is the discovery cohort, and then they validated their findings here. This is healthy donor, benign pancreatic disease. This is precancerous pancreatic lesions and pancreatic ductal adenocarcinoma. And as you can see here, they are showing 100% specificity and 100% sensitivity. So if this is validated later on in a larger cohort of pancreatic ductal adenocarcinoma, then this will be a very good marker for early detection. They also showed that if you have a lower level of circulating exons with which are glypican positive, then you have better survival as compared to the one that has higher circulating exosome that are glycosome that are glypican positive, but they do not find that in the CA19-9. So again, they are saying that it is also a prognostic marker in the pancreatic cancer patients that how well they are going to do. I mentioned a little bit about the molecular makeup, that there are different molecular subtypes of the pancreatic cancer that respond differently to treatment, and they have different prognosis. And these are some of the examples. So one of the studies by Bert Vogelstein lab at Johns Hopkins, what they did, they did the genomic analysis of only 27 tumor sample. This study was done a long time ago, 2008. And what they found that each tumor had at least 68 or 70 different genetic alterations. And these all genetic alterations corresponded with 12 core signaling pathway that was altered. But at the same time, what they also found that if you compare two tumors, there were different genes that were altered in each of these different pathways. So the summary of this slide is that it's highly heterogeneous tumor. And we see a lot of heterogeneity, intertumor heterogeneity in this cancer. And then in 2011, another study was published in which they did the gene expression analysis of pancreatic ductal adenocarcinoma. And based on the gene expression, they divided these into 3 different subtypes. And one of the subtypes, which is here, quasi-mesenchymal subtypes, had the poorest survival among all these pancreatic cancer. On the same line, in 2015, there was a study where they analyzed the variation in chromosomal structure, and they identified 4 different pancreatic ductal adenocarcinoma subtypes: stable, scattered, unstable, and locally rearranged based on the degree of chromosomal alterations. And the unstable type was the one where the most chromosomal variation occurred. And this was also associated, this subtype, with the alterations in the DNA repair pathway. And the patients which represented unstable subtype, they responded very well to the platinum therapy. And they correlated that because they have alterations in that DNA repair mechanism, that's why they responded very well to the platinum drug. So that's how these are providing proof of concepts that the patients, for example, stage 1 or stage 2 patients, they should not be treated that they all are same patients, but they can be subgrouped based on their genetic molecular makeup and select and stratify those patients that will respond differently to a specific drug. And then again, last year, there was another study in which they virtually microdissected using an computer algorithm where they separated the epithelial tumor cells gene expression and the stromal gene expression. And based on the stromal gene expression, so they divided into the activated stroma and the inactivated and the normal stroma. And what they found that no matter what the genetic makeup of the tumor is, if the stromal tissue shows the activated stromal gene expression, then the survival was poor. So what they were trying to say, that the studies where we take out the patient tumor and do the xenograft studies in the mice may not represent the actual tumor of the patients, because if the stromal gene expressions are active stroma, then it will respond differently to the treatment. And again, recently in 2016, so you can see this is a highly active area to find out the different subtypes in the pancreatic tumors so that they can improve treatment. So this is the largest study, almost 500 patients, and they described 4 different subtypes, and they were overlapping with the previous studies describing different subtypes. And then based on the metabolism and the metabolic activations, this was again divided into slow proliferating type, glycolytic, and lipogenic. And this is the last study on the molecular subgroups of pancreatic cancer. So talking about the difference in metabolic programming or metabolic characteristics of pancreatic tumors, what studies have found that pancreatic tumor cells, they reprogram their metabolic pathways. And this is a highly complex slides, but I would just highlight 3 points. And one is that you have increased glycolysis here. You also see the glycolysis intermediates that goes into the non-oxidative segment of the phosphate pathway here. And there is an alternate glutamine metabolism here, which is unique to the pancreatic cancer, where glutamate is converted to aspartate, catalyzed by GOT2. And this aspartate is shunted out to the cytoplasm and gives pyruvate through several sequential steps. And during this process, NADPH that is produced is used for the redox balance instead of the NADPH that is used by the pentose phosphate pathway here. So this is not active here, rather it has an alternate metabolism. So these are the 3 points: increased glycolysis, alternate glutamine metabolism, and the activation of the non-oxidative pathways. These are the 3 important points. And many of these enzymes that are catalyzing these reactions are modulated by KRAS. So if you remember, about 95% of the pancreatic tumors have KRAS mutations. And then recently, last week, in August, a couple of months ago, what they found is that, as I mentioned, that stroma is very helpful for pancreatic tumors. And one of the important components in the stroma is the pancreatic stellate cells. And these stellate cells, in fact, produce alanine, and it supports cancer cells. It fuels TCA cycles, supports lipid biosynthesis, and shunts glucose to serine and glycine biosynthesis. So this is the latest concept in pancreatic cancer metabolic rewiring, where the pancreatic stellate cells in the stroma can support and there is a positive feedback loop. What we don't know, that what makes tumor cells undergo autophagy to produce to activate pancreatic stellate cells to produce alanine. So let's talk a little bit more about stroma, because these are some of the latest progress that have been made in pancreatic cancer. So what has been found, this is also a complex slide, but I will make it highly simple. And you see here the environment is immune suppressive. So CD8 positive T cells is inhibited by Treg cells, MDSC, and tumor-associated macrophages. That is the one part. Pancreatic stellate cells, which I just mentioned, that enhances the tumor stroma, which is called desmoplastic reactions. And then, as I mentioned briefly, that due to the highly desmoplastic stroma, you have accumulation of hyaluronic acid that increases the interstitial fluid pressure on the blood vessels, and that inhibits the drug flow to the tumor. Okay. So how we can target a stroma to enhance drug delivery? So there are two studies that have been done, and this is the mouse model that have been used in this study. This mouse model has pancreas-specific activation of KRAS and inactivation of p53, and they developed pancreatic ductal adenocarcinoma, which exactly recapitulates the development and progression of pancreatic cancer in human. So they use these models, and what they found is that if they inhibit the stroma through inhibition of Hedgehog signaling pathway, then they increase the survival in this mice with pancreatic cancer, and they also decrease the liver metastasis. Then there was another study here where they reduce the hyaluronic acid deposition in the stroma by using hyaluronidase, and that reduced the interstitial pressure, and the blood vessels that were compressed got relaxed, and there were more drug delivery in the tumors. And they found that by doing this, they enhanced survival, and they also reduced metastasis. So this approach is currently under phase 2 clinical trial, and they have some positive responses. But there are two other studies that contradicts these findings by saying that removing a stroma or inhibiting a stroma may not always be beneficial. So in these two studies that were published back to back in Cancer Cells, what they shown using a genetically engineered mouse model, that if you inhibit sonic hedgehog and reduce the stroma, in fact, the survival goes down. So reduced survival here, and then you also see increased metastasis, and you see increased the precancerous lesions. Then in another study on the same line, published in the same journal, back to back, they showed the same thing, that myofibroblast depletion enhanced pancreatic ductal adenocarcinoma. So myofibroblasts are the stromal cells in the desmoplastic stroma, and when you inhibit those myofibroblasts, you see increase in the tumorigenicity, and you also see decreased survival. So based on these contradictory studies, I think more work needs to be done, and more to identify that which patient will respond better by anti-stromal therapy. So you have to reprogram a stroma in order to enhance sensitivity to the drugs. So tumor-stromal interaction is a complex and therapeutic approaches. Targeting a stroma needs caution and may require new molecular taxonomy in pancreatic cancer. So let's talk about one of the glycoprotein that is highly expressed on different tumor types. And this is mesothelin. This is a glycoprotein that was discovered right here at NCI in Ira Pastan's lab. And this was targeted by using immunotoxin in mesothelioma, which very successfully, with very good results. So this slide is just to show you that pancreatic cancer, along with other cancers, also express large amount of mesothelin. So this is a good target. So what Sunil Hingorani and groups did, which they re-engineered T cells here to target mesothelin, and they did this, and this worked very well by penetrating through the very dense stroma and also immune suppressive environment, and in fact they lysed tumor cells by binding to mesothelin, and also enhanced survival in these mice. So this is also going to go in the clinical trial, and let's hope that that something comes up that will be beneficial to the patients. This is the highly advanced model that has been developed recently, just in last 1 year, which is called organoid model. And David Tuveson at Cold Spring Harbor Laboratory developed this model, and this is going to be the most important preclinical tool, both to understand individual tumor biology and also identify drug targets and test different novel targets. So what happens that from the patients, they can take small part of the tumors, and they can develop in outside in the culture, 3D culture, exactly the same architecture of tumor as you see in the patients, and this can be frozen. And after that, if you transplant that in a mouse model orthotopically, it starts with the development of precancerous lesions and going to the advanced cancer. So this is one of the latest model. I just wanted to tell you about this. How much time do you have, Terry? Okay, great. So I will take 10 more minutes, and please let me know if any of these points what I'm trying to make is not clear, so I can stop and then explain to you. Inflammation is an important component in pancreatic cancer development and progression. Chronic pancreatitis, as I mentioned, enhances the risk of pancreatic cancer. There are several oncogenic pathways that are constitutively active, for example, NF-kappa B signaling, desmoplastic stroma, huge stroma, highly active inflammatory cells, and inflammatory cytokines, COX-2 and NOS2. These are the two enzymes that catalyzes different reactions that produces reactive species. So if you look at the schematics of normal pancreatic intraepithelial neoplastic lesions, precursor lesions, and PDAC, you see a stepwise accumulation of inflammatory changes here that accumulates over different stages. There are also evidence which shows that, as I mentioned, KRAS mutation is very important. It activates KRAS, and more than 95% of the patients have KRAS mutation, but all the mutated KRAS is not tumorigenic. So what has been shown that mutated KRAS has to reach a pathological level of activation, and that is provided by these reactive species that are generated during chronic inflammation. Just an example. So one cytokine that we are working in my lab. So I will give you a brief overview with 4 or 5 slides that what we are doing in our lab here at NCI about pancreatic cancer. And we are interested in one of the cytokines that is called macrophage migration inhibitory factor. It's a pro-inflammatory cytokine expressed in epithelial and inflammatory cells. It was described as regulator of immune response, increased expression in tumors. It also activates oncogenic signaling pathways, NF-kappa B, AKT, ARK1/2, enhances some of the enzymes which I just mentioned. It also antagonizes P53RB2F pathways. And based on these and other functions, it is described as a link between inflammation and cancer. So we tested a simple hypothesis. MIF contributes to pancreatic cancer progression and predicts disease outcome. These are the tumor and adjacent non-tumor tissue. You see increased expression of MIF in the tumors. These are the normal pancreatic duct. When you do the survival analysis, this is the Kaplan-Meier analysis. And if you group the patients in two groups based on the MIF expression level as high and low, based using median value as the cutoff, then you see that the high MIF expressing tumors patient have poorer survival than those patients where tumor expression is relatively lower. And this can be validated in several different groups of patients or the cohorts and also the publicly available data sets. So this simply shows the association, what MIF actually does. So we did an in vivo experiments in which you overexpress MIF and transplant those cells in the tumors. You see that MIF expression tumors was larger and also more metastasis here. When you look at the tumors histology, they are completely different. The control tumors showed highly differentiated or moderately differentiated or well differentiated, but the MIF expressing tumors are poorly differentiated, that are more aggressive. When you look at the gene expression analysis between these two tumors, you see a change in the global gene expression profile. And some of these genes represent that the EMT pathways, and MIF overexpressing tumors showed expression of EMT marker genes. Then in this paper, recently in Nature Cell Biology, David Leiden's group showed that in the pancreatic cancer patients, if the circulating exosomes have higher macrophage inhibitory factor level, then they are more prone to metastasis. As I mentioned earlier in my talk, that pancreatic cancer patient, earlier stage patients, some metastasize faster, some don't. So maybe be those patients who have high MIF expression, they are at increased risk of liver metastasis. So we wanted to see that what is the difference between the tumors who are expressing high MIF level and those that are low MIF level. So we asked two questions. What are the molecular distinctions between these two groups of tumors, and what are the mechanistic and functional role of MIF in tumor progression? So a highly talented postdoc in my lab did the integrative transcriptomic analysis in which he analyzed these two groups of tumors by mRNA and microRNA arrays. And what he tested the hypothesis that MIF regulates microRNA associated with tumor progression and disease aggressiveness. And he found several differentially expressed microRNA between these two groups. Some of them were also associated with survival. He validated these data in multiple cohorts of pancreatic ductal adenocarcinoma. And what we found at the end, that MIF enhanced a microRNA, MIR301B, and MIR301B targeted one of the potential tumor inhibitory gene, NR3C2, which has not been described in pancreatic cancer before. And this NR3C2 was inhibiting EMT. So if you have increased MIF, it enhances MIR301B, and 301B inhibits NR3C2, then there will be more EMT and there will be more progression and metastasis. So the hypothesis is now that MIF NR3C2 signaling is a potential therapeutic target in pancreatic cancer. So we went further and we used the same model, and we deleted the MIF from this KPC mouse model, which I earlier described. And what we found, that when you removed MIF genetically from these mice, it significantly enhanced survival, you can see here. And it also decreases metastasis in these mice. And then we did some molecular studies, and you found that MIF-deleted mice, they had increased E-cadherin, decreased ZEB1, vimentin, and N-cadherin, and these are EMT marker. This is Western blot analysis. And then again, you see reduced microRNA 301B expression in MIF deficient KPC mice, and increase the NR3C2 expression, just to show that what we are showing, finding in the human tumors, the same we are finding also in the disease model. So now it's a potential target. So how we can go further? The further is that now use the MIF specific inhibitor, because you can't delete gene in the patients, so you have to use these inhibitors once the mice develop this tumor. So that's what we are doing. There are several ways. You can use a small molecule MIF antagonist, which we are using. We are also using anti-MIF antibodies, which is in clinical trial for other cancers. And in summary, from this last experiment which I showed you, is that a higher MIF expression is associated with poorer outcome. MIF enhances growth and metastasis of tumor xenografts in mice. MIF-driven signaling inhibits NR3C2, which is an it's a tumor suppressive, potential tumor suppressive genes by upregulating MIR301B. And NR3C2 is a negative regulator of EMT. MIF deficiency increased survival and reduced metastasis. Therefore, MIF, MIR301B, NR3C2 signaling is a potential therapeutic target. So I would like to end with this slide, which I showed right in the beginning, that in order to achieve early detection and effective therapy, we need to understand extensively pancreatic tumor biology. And in last 6 to 7 years, there have been enormous amount of effort that are made in understanding pancreatic tumor biology. And now we are at a point that we have several leads that can be taken into the preclinical and clinical studies. So I will stop here and take your question if you have any. Thank you very much for your attention at 5:30.
So does MIF have any effect on the immune system?
Yes, it does. So what happens that MIF enhances the level of MDSC and the level of T regulatory cells in the tumors. So when we check the tumors, MIF expressing and control tumors, we found that when you remove MIF, you have less of those immune suppressive cells in the tumors.
Okay, that'll do it.
Great. Thank you very much. Thanks.